All right, pyruvate oxidation is the part of cellular respiration that moves all the reactions into the mitochondria.
We know by now that glycolysis takes place in the cytoplasm and leads to the formation of 2 molecules of pyruvate per molecule of glucose. These pyruvate molecules are converted into acetyl coenzyme A, also known as acetyl-CoA, and this happens inside the mitochondrial matrix. Therefore, acetyl-CoA is the key metabolic intermediate that links glycolysis and the citric acid cycle. For clarity, we will not show the coenzyme portion of acetyl-CoA, and instead are simply representing it as the 2-carbon molecule acetate. So how does the pyruvate get into the mitochondrial matrix? This depends on a carrier protein that transports pyruvate into the mitochondrial matrix, where there it is oxidized to form acetyl-CoA by the enzyme Pyruvate Dehydrogenase. In this reaction, a molecule of carbon dioxide is generated as pyruvate is oxidized to acetyl-CoA, and a molecule of NAD+ is reduced to NADH. This last reaction is not shown for clarity. The citric acid cycle consists.
This is actually a really good video. He goes into a lot of really good detail, and it's supposed to look a little bit more quote-unquote lifelike by giving the three-dimensional shape of all these proteins. But we are only going to watch this first part. And if you noticed, he said a carrier protein brings pyruvate into the mitochondria. So that was on our last test, transport protein. And then he also noted that carbon dioxide is produced and NAD plus is reduced, which means it gains electrons. So it's the electron carrier, and NADH is produced. So let's go back to our notes. The prep step occurs. It begins in the cytoplasm. It ends inside the matrix. The starting materials were the pyruvate from glycolysis, and the products are the acetyl-CoA molecules, which are going to be the molecules that go into the citric acid cycle. No ATP is produced here. However, CO2 is produced, and this is the first stage where CO2 is produced. And remember, that's significant because glucose will eventually be broken into 6 molecules of CO2, 6 molecules because glucose has 6 carbons. Are the electron carriers involved? Yes. NAD plus does get reduced and then takes those electrons to the end of cellular respiration. Special information would be the fact that it is taking place in the cytoplasm and then in the mitochondria. It's the only step that is located in both regions of the process. All right, next to the citric acid cycle.
The citric acid cycle is a series of chemical reactions that takes place in the mitochondrial matrix. This cycle uses acetyl-CoA derived from sugar and fat breakdown to form ATP, NADH, FADH2, and carbon dioxide. The NADH and FADH2 can be used to form additional ATP through the electron transport chain. The citric acid cycle goes by many names, including the tricarboxylic acid, or Tca cycle, and the Krebs cycle.
This is one of those places that biology is incredibly annoying. The citric acid cycle, you will also see written as the Tca cycle and the Krebs cycle. They will be used interchangeably. You're expected to know that they are all the same thing. And I don't want to tell you that photosynthesis has the Calvin cycle. Yes, we have another cycle, and it starts with a C, and it really couldn't be called like the frog cycle or something that was totally different. And that is going to be confusing once we get to photosynthesis, so we're not going to worry about it right now, but I'm still bitter, and that's why I'm bringing it up. But citric acid cycle is part of cellular respiration, also called the Tca cycle, also called the Krebs cycle.
Citric acid refers to the citrate that is produced in the first step of the pathway. The tricarboxylic acid title gets its name from the 3 carbon dioxides that are produced for each fully oxidized pyruvate. Krebs refers to Hans Adolf Krebs, who identified the full cycle in 1937. He was eventually awarded the Nobel Prize for Physiology or Medicine in 1953 for his discovery. In the first step of the cycle, an enzyme called citrate synthase joins the 2-carbon acetyl group from acetyl-CoA with the 4-carbon oxaloacetate to form a 6-carbon citrate. In step 2, an enzyme called aconitase converts citrate into isocitrate. Next, an isocitrate dehydrogenase enzyme oxidizes isocitrate, a 6-carbon molecule, into a 5-carbon alpha-ketoglutarate. The carbon that was lost is released as carbon dioxide, and 1 NADH is also formed. The carbon dioxide that is released was originally part of oxaloacetate and not acetyl-CoA. In the fourth step, an enzyme called alpha-ketoglutarate dehydrogenase converts alpha-ketoglutarate into a 4-carbon succinyl-CoA. Similar to step 3, this reaction produces 1 carbon dioxide and 1 NADH. In step 5, a succinyl-CoA synthetase enzyme converts succinyl-CoA into succinate. This produces GTP, which is converted to ATP. In step 6, an enzyme called succinate dehydrogenase converts succinate into fumarate. This step makes 1 FADH2. In step 7, a fumarate hydratase enzyme then converts fumarate into malate. In the final step of the citric acid cycle, a malate dehydrogenase enzyme converts malate back to oxaloacetate. Like all steps involving a dehydrogenase, a coenzyme is produced. Here it is NADH. The oxaloacetate that was regenerated through the citric acid cycle is now ready to join with another acetyl group and begin the cycle a second time. For every 1 glucose that is broken down through glycolysis, 2 pyruvates will be produced. These 2 pyruvates will produce 2 acetyl-CoAs. So for every 1 glucose, 2 acetyl-CoAs will be made and 2 turns of the citric acid cycle will occur. This means each product of the cycle must be doubled. A total of 4 carbon dioxides, 6 NADHs, 2 FADH2s, and 2 ATPs are made through the citric acid cycle. NADH and FADH2 are electron carriers that can produce more ATP later in aerobic respiration. In addition to sugars like glucose, proteins and fats can also provide carbon substrates to fuel the citric acid cycle. Proteins can be broken down into individual amino acids such as alanine, aspartate, and glutamate, and converted into intermediates in the cycle. Fatty acids can be broken down into acetyl-CoA, which then begins the citric acid cycle. This metabolism of sugars, proteins, and fats through the citric acid cycle provides the vital energy necessary to maintain many cellular processes.
All right, let's go back. Whoops. Yeah.
NADH and FA of the cycle must be doubled. A total of 4 carbon dioxides, 6 NADHs, 2 FADH2s, and 2 ATPs.
Of course, you don't need to know all those molecules and enzymes, and they're not really that hard to memorize. If you notice, a lot of them were very, very similar. So if you take biochemistry, you will have to learn this, but it's not as bad as it looks. But what we're concerned with are the products, if CO2 is produced, if ATP is produced, and if the electron carriers are involved. Notice that 4 molecules of CO2 are produced here, and she pointed out that the citric acid cycle will run twice for every molecule of glucose, and that's because glucose is broken into 2 pieces during glycolysis. So we have the 2 pyruvate molecules that enter the mitochondria. So be careful with the questions on the test. You have to be really careful to see if the question is asking you about 1 pyruvate molecule or 1 glucose molecule. For glucose, there's going to be 4 CO2 molecules produced, and including the 2 that were produced during pyruvate oxidation, glucose is completely oxidized by the time the citric acid cycle has run twice. Notice we have the electron carrier NADH is reduced here. And then we also have a second electron carrier, FADH2, that is only part of the citric acid cycle and not anything else. And then we have ATP produced again. And as she said, NADH and FADH2 will go to the last stage of cellular respiration and help generate even more ATP. So let's look at our study guide. All right, citric acid cycle, where does it occur? In the matrix of the mitochondria, and the matrix is the cytoplasm inside of the mitochondria. The starting materials were acetyl-CoA. The products are carbon dioxide, NADH, FADH2, and ATP. Is ATP produced? Yes. A total of 2 molecules of ATP are produced here per 1 molecule of glucose. So we have 2 molecules of ATP produced in the citric acid cycle, and we had 2 molecules of ATP produced in glycolysis. Are the electron carriers involved? Yes. Both of the electron carriers are here. CO2 is produced, 4 molecules of CO2 per molecule of glucose. Add that with the 2 molecules of glucose produced during the prep step, and glucose has been completely oxidized into carbon dioxide. Special information, the fact that this is where glucose is completely oxidized into CO2. You could be asked a question where that occurs, and then the answer would be the citric acid cycle. All right, the last stage of cellular respiration is oxidative phosphorylation.
Our body is made up of trillions of cells. They all require energy to function. This energy is created within our cells, in the mitochondria. Here, food is converted into chemical energy called ATP. ATP is released by the mitochondria so cells can use it. Mitochondria consist of 2 membranes, an outer membrane separating it from the cytosol, and an inner membrane surrounding the so-called matrix. The area between these membranes is called the intermembrane space. ATP is generated at the inner membrane of mitochondria by an efficient mechanism called oxidative phosphorylation, involving several membrane protein complexes. Nutrients provide high-energy electrons in the form of NADH, which are used by the protein complexes to pump protons from the matrix to the intermembrane space. This continuous pumping creates a proton gradient, where the positively charged protons are attracted to the more negative matrix. When the protons re-enter the matrix through the ATP synthase protein complex, they catalyze the production of ATP.
All right. Now, I liked this animation. They have a lot of good ones. There's a lot of really good videos about all of this. Now, I like this one because it was short. And they do a really good job illustrating what is happening in the inner membrane, also known as the cristae. So we have the electron transport chain, and then we have ATP synthase. And as she explained, the electron carriers bring electrons to the electron transport chain. As the electrons are transported through the electron transport chain, the electrons lose energy because the electron transport chain, these proteins use that energy to pump protons into the inner membrane space. So this is active transport because it uses energy. It's a common mistake to think that active transport has to use ATP. It doesn't have to use ATP, it just needs energy, and ATP is one way to get energy. But these electrons are providing the energy, so the protons are being pumped against their concentration gradient. And the electron transport chain, the job of the electron transport chain is to make this gradient, because this gradient is going to power ATP synthase. ATP synthase cannot phosphorylate ADP unless protons are moving through the channel. So if there's no gradient, then there's no flow of protons through that carrier protein, and no ADP is phosphorylated. Okay, so what happens is we have the protons being pumped to the inner membrane space. So we see that this concentration is increasing. And chemiosmosis is this proton gradient. And so the process that occurs in ATP synthase is a result of the proton gradient that the electron transport chain sets up that proton gradient, and then the flow of the protons by passive transport will turn on the enzyme in ATP synthase, and then that will phosphorylate ADP. And these two processes together are oxidative phosphorylation. It's a common mistake to think just the chemiosmosis, just ATP synthase is oxidative phosphorylation, or just the electron transport chain is oxidative phosphorylation. It's both. You have to have both, because the electron transport chain, it sets up the gradient that is necessary for ATP synthase to function. You can kind of think of it as an on and off switch. All right, let's go back to our study guide. Okay, oxidative phosphorylation. is composed of these two processes. Electron transport chain, where does it occur? These proteins are they're embedded in the inner membrane of the mitochondria, also called the cristae of the mitochondria. Now, out of the context of this test that is only covering enzymes and cellular respiration, if you were taking a test that included photosynthesis, then you would need to be very clear about the fact that this electron transport chain is in the membrane of the mitochondria, because I hate to tell you, but there's an electron transport chain in the membrane of the chloroplast during photosynthesis. So I didn't want to have to tell you that yet. So on the next test, you're going to have to be very clear about where the electron transport chain is. So the starting materials would be the electron carriers, and they are dropping off the electrons. So the products of this process would be the proton gradient. And then also oxygen is going to be the final electron acceptor at the end of the electron transport chain. So when oxygen grabs onto those electrons, it will grab hydrogen as well and form water. So technically water is also a product of the electron transport chain. During the electron transport chain, technically ATP is not produced. But you have to be careful because some textbooks, etc., will call the whole process electron transport chain. So I will not get that nitpicky with you. CO2 is not produced because CO2 has already been produced. Glucose is completely broken down, so there's no source of carbon for carbon dioxide to be produced. Are the electron carriers involved? Yes, they are dropping off the electrons. Special information would be that the electron transport chain is setting up the proton gradient that will power chemiosmosis and the phosphorylation of ADP. So chemiosmosis is also embedded in the membrane. ATP synthase is the transport protein / enzyme that will move protons from the inner membrane space into the matrix by passive diffusion. And the products are ATP. So is ATP produced? Yes, a lot. A lot of ATP is produced. Like 34-ish molecules of ATP, depends on where that cell is. And CO2 is not produced here either. All the CO2 has been produced earlier. The electron carriers are kind of involved because they set up the electron transport chain. Technically, they're not interacting with ATP synthase, but like I said, I'm not going to get that nitpicky with you. And special information, this is where most of the ATP is produced during cellular respiration. And remember, cellular respiration, the purpose is to phosphorylate ADP. Substrate level phosphorylation, that is a way to phosphorylate ADP that took place in glycolysis and the citric acid cycle. And then the second type of ADP phosphorylation is oxidative phosphorylation. And this is where oxidative phosphorylation occurs at the very end of the whole process. So now we're going to watch that original animation again. And hopefully it will be even more familiar since we just watched it about 20 minutes ago.
that bite of food into a chemical that a cell can recognize and use as energy. The first step is altering the food into its component chemical compounds and then getting those molecules into your cells. That process is called digestion. Once inside your cells, the process of turning that bite of food into useful energy by cellular respiration begins. The process of digestion results with carbohydrates and other molecules being removed from the consumed food and transported into the bloodstream. From there, nutrients, like the carbohydrate glucose, will leave the bloodstream through a capillary wall and enter a tissue cell. Once inside the cell, cellular respiration will completely oxidize the glucose molecule, releasing high-energy electrons. The overall goal is to make ATP, a storage form of energy for most cells. Cellular respiration is a four-stage process that begins with glycolysis. Glycolysis literally means splitting sugars, and it is the first step of cellular respiration, occurring in the cytoplasm of the cell. Glycolysis consists of two distinct phases, an energy investment phase and an energy harvesting phase. In the energy investment phase, two ATP molecules transfer energy to the glucose molecule, forming a 6-carbon sugar diphosphate molecule. This molecule splits, and the energy harvesting phase begins. During this phase, the two 3-carbon molecules are converted to pyruvate, and ATP is formed. Glycolysis is a 10-step reaction that involves the activity of multiple enzymes and enzyme assistance. In the process, a net of 2 molecules of ATP, 2 molecules of pyruvate, and 2 high-energy electron-carrying molecules of NADH are produced. When oxygen is present, the pyruvate molecules and NADH enter the mitochondria, and the next stage of cellular respiration begins. The next stage of cellular respiration involves the movement of pyruvate into the mitochondria, where it undergoes oxidation. Each pyruvate molecule is converted into a compound called acetyl-CoA. In the process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and a carbon is lost, forming carbon dioxide. The next stage is the citric acid cycle, also called the Krebs Cycle. Here, acetyl-CoA will bind with a starting compound called oxaloacetate, and through a series of enzymatic redox reactions, all carbons, hydrogens, and oxygens in pyruvate ultimately end up as carbon dioxide and water. The pathway is called a cycle because oxaloacetate is the starting and ending compound of the pathway. For every glucose that enters glycolysis, the cycle completes twice, once for each molecule of pyruvate that entered the mitochondria. During pyruvate oxidation and the citric acid cycle, a net of 8 NADH, 2 FADH2, 2 ATP, and 6 CO2 are produced for each glucose molecule. In order to understand how the majority of the energy is produced by aerobic respiration, we need to follow the NADH and FADH2 molecules to the next stage, the electron transport chain. The electron transport chain is a series of membrane-bound carriers in the mitochondria that pass electrons from one to another. As the electrons are transferred between the membrane proteins, the cell is able to capture energy and use it to produce ATP molecules. Proteins in the chain pump hydrogen ions across a membrane. When the hydrogen ions flow back across the membrane through an ATP synthase complex, ATP is synthesized by the enzyme ATP synthase. Oxygen acts as the terminal electron acceptor. By accepting electrons, oxygen is reduced to form water, a byproduct of the electron transport chain. All the high-energy electron carriers from the previous stages of cellular respiration bring their electrons into the chain. From this, the bulk of ATP from the entirety of cellular respiration is produced. A net of 32 to 36 ATP. In summary, we have seen how the 4 stages of cellular respiration are responsible for converting the energy found in the glucose molecule into ATP, the energy battery of the cell. On average, 36 ATP molecules are produced per glucose molecule that entered the cell. In the process of producing ATP, oxygen is brought in from the bloodstream to be the final electron acceptor in the electron transport chain, and the carbon dioxide that is produced as a byproduct is released. The goal of cellular respiration is to transfer the energy from the food that we eat daily into ATP that our bodies can use.